Hi, every good book deserves good illustrations, and sometimes there are even useful, and you cannot spoil the good book anyway. In this video I will talk about illustrations for DESeq2, and I will mention two sources. One is DESeq2, which is online tool for gene expression analysis, and another one is DESeq User's Guide, which is an official user's guide in bioconductor, and if you scroll far enough you will come across some figures which are quite useful. Obviously, before you do illustrations you need DESeq2 script. Last time I explained you how you can generate a DESeq2 script using DESeq2 online tool. This button is available for some but not for all RNA-seq data. The principle for selection, I guess, is the relatively simple composition of the dataset. This one, for instance, consists of paired samples of a baseline and a metformin treated patient. And by pushing DESeq2 button you enter online analysis of these samples. On this page you select groups and push analyze button, and after a while you get the table of results and the figures. This set of figures resembles what you have seen with DESeq2 using Lima. It has a dispersion plot different from what you have in HR, but probably useful for technical assessment. Then this is pretty useless distribution of p-values. Sometimes it's shifted more to the left, more to the right. It means how many significantly different genes found. In this particular case it's only one. This figure is for nothing because you can simply say 1 instead of drawing such a figure. This is important. Box plot is a fast and easy check for compatibility of samples. Here we can see that these two groups are comparable and you don't see any obvious outliers. Looks okay. The UMAP is controversial because UMAP usually used for really big data sets, and this data set is not big at all. You better use MDS or PCA plot instead. And these two figures, the last one, it's two ways of demonstrating differentially expressed genes. One is in a scale of log2 fold change and minus log p-value, so it goes up if p-value goes down. So this is the one and only differentially expressed gene which is significant, and in this scale you have a x projection of from low to high expressions and again fold change, and this lonely gene is RMD5A. And of course you have a R script attached which you copy paste into your RStudio, and you can modify and improve it further. Working with a single differentially expressed gene is not very handy, especially if you want to demonstrate figures. Therefore, I took another data set for today. It's another RNA-seq data, which also has a button DESeq2. As you can see, it has 3 groups, 2 cell line controls and 1 cell line treated with some stuff. Details you can read further if you're interested in obesity and adipose tissue. But for us it is important to check the script and illustrations to it. I made this script online in DESeq2, then copy-pasted into RStudio, and in its native format you do the DESeq2 library, then you import the data. Pay attention that you cannot get this read counts file from a usual place of supplementary files. Here you see some summaries which are not what you need. In other words, the path which you generate here is the only address where you can get the row counts data, You can copy paste it into web browser and you will download it into your download folder, or you do it this way, that's equally okay. Then you load annotation as usual. Then there is no sample information. Instead you have this selection which you do online. It's certainly not good for the RStudio script if you work offline. Then it is a tricky part of the creating sample table based on the order of the samples in the groups, then naming them, then declaring them all as factor with the levels. That's important. It's not just a data frame. Then you do a filtering. Then finally you come to these two major DESeq lines. One is declaring variable, which includes count data, sample info, and design all in one line. Then you do analysis. Then you pick the results. I skip details on purpose, although you can ask for results. And it says that it is a CM control versus control, which is this group and this group. Then there is this standard cut with 250 genes, which is also not good. There is no tops here. It's a full list of TT, not a full list but 250. Then you can write it. And then the very first plot comes here, which is dispersion and histogram. And then volcano plot, MD plot. It's just a plot composed of columns from the results table. Then Venn diagram, which you don't need, and box plot, which is far too late, so it has to be improved. Let's modify this script in such a way that we can work entirely in RStudio. As I said, path is already the link to the download folder, and therefore you either get it from online this way or you download it, which I did, and read it with the read CSV. It's equally good. Then I keep this annotation since it is available, no problem. There was a problem with the sample information, although, and therefore I use a GeoQuery library and get the information from the site about this dataset. And as usual, you have here many different data, but these are all about samples. And so you copy paste address and get the sample info, and check the column names and decide what columns you take, and it will be this one. And so this is the major group, the third one, and these are described as the two controls. Pay attention that in this particular case the column names are in a good order from 13 to 21. So here I partially inherited the way how it was made with Geo2R, because I need factor and I need group names, which I agree, and then I need levels, and then I also found the batch information and added to this sample file, simply declaring it. And now if I ask for a structure of sample info, I see that it is 3 levels each, marked like this. That's okay, that is acceptable. Then I do filtering as usual, and now, actually already now, I better have a look at this data using a box plot. I use the suggested color palette, why not? And then I can enjoy watching a box plot. And you see immediately that this first group is not good. It is a much smaller distribution. The boxes are smaller than here, and this is where you can foresee the first problem coming. And you can also add legend, why not? and I explicitly changed the xlim to give space for the legend, otherwise you wouldn't have it. Then you do DESeq dataset. Pay attention, I added 0 to the design to make my selection of groups easier. It doesn't influence the results. Then you can do PCA plot out of those data, but before that you have to do a transformation, the VSD-RLD for this and other figures. What is it exactly? Please read instructions. And PCA plot is like this. And again, you see the problem with the group number 1. It's horribly far away from two others. So in addition to some biological differences, this seems to be a batch effect. Then, before any further analysis, you can compare samples using heat map. Before that, you do a sample distances measurement and distance matrix. And the heat map, it's a bit confusing because it's a bit inverted colors. So this is highly similar samples of group 1, and these are highly similar samples of group 2 and 3, and these distances are very far. And it agrees with what you have seen on PCA plot. Then we import VCN library and plot mean SD plot, which is standard deviation versus rank mean expression values before any trends and dispersion analysis. It can be useful for you or not, it's up to you to decide. Now we do the analysis, everything in one line. Now we do another dispersion plot, no further comments. And next we come back to a box plot. It has two options, normalized and not. This is not normalized, and now we can repeat it. Normalized, and you see that even after normalization, these boxes are smaller than those two groups, which is obviously a batch effect. And therefore, for the results, we select group 2 and 3, so we compare this group and that group. And if you ask, can I be more flexible in selecting groups and what other groups in general, then you can call this function results names, ds, and it tells you that there is a group control, there is a group CM control, group CMCC, and batch 2 and 3, batch 1 is missing. It just as usual, that second factor in your model is always truncated. And so you can call contrasts like this. So first you say what factor it is, group in this case, and then you call by name CMCC and CM control. Or if you want to compare batches, simply check is there any difference in batches. You similarly say batch is a factor, and then what is remaining is 2 and 3. And by the way, if you compare batch 2 and 3, there is no difference between them. And then you come to the same results, and contrast is the contrast in this case. Right, now we finalize the results into a table and generate the tops. And tops are already annotated with the gene symbols, no problem, and you can write it. Now the remaining figures out of the GeoTOR histogram of p-values, I wouldn't use it. Volcano plot, you can use it. If you like this design. There are better volcano plots, by the way, I will come to it in other video. Then MD plot, which is other way of showing the same differential expression results in another scales. And authentic mdplot is plot MA in DESeq tutorial like this. So you have two. Venn diagram I would skip, and then UMAP we don't need it, and at the end you can do heat map of differentially expressed genes in your tops, but pay attention, the total number is far too big and you better do only a part of it. So I take first 50. And as a rule, heat map doesn't like too many names. What is useful in a p-heat map that for annotations you take sample info table which generates those bars and those extra annotations. So it is very suitable for differential expression visualization. And another useful plot if you want to validate a difference in one particular gene. In this case I took EF27. I simply use the information of the tops again. Here EFI27. That's what my mistake. And so it is. These are almost all illustrations you could find and use from Geo2R and DESeq2 user's guide. I skipped a few. You better not take all of them, but be selective. Take what you find appropriate and suitable for your presentation or publication. That's it.